welcome everyone here in the department, and also welcome everyone that's online with us on Zoom. So you are going to see me a few times because all my students will actually have to present their seminars this semester. So I have to remove, the old jacket from the wardrobe and all of that. But today we have Mr. Jonathan Stephen Cardenas Gallegos. So you might not know, but he is officially my first UGA student. Actually, the first one that signed the offer letter. So you probably didn't have that student. So
Will he be the first one to finish?
I hope so. So first back to back, yes. But the satisfaction of being here presenting Jonathan is immense because Jonathan is born in Ecuador, and he did his bachelor's in the same area that I did, agronomic engineering, and had developed actually a tremendous passion for precision agriculture. But before that, he liked horticulture. He worked with Ecuador's probably most famous crop, horticulture crop, which are roses. He did an internship in ornamental crops, right, Jonathan, and got his feet into horticulture, and then was offered a position in the Czech Republic to work with high throughput phenotyping. That put him in one of the most prestigious lab in this field at the moment. And since then, Jonathan started trying to solve complicated problems by using all his skills. This is what we're going to hear him talking today. Jonathan already told me that he likes a lot of things, and his list was so long that we would have a seminar about everything that he likes doing. But the person behind the researcher really like, you might know that famous juice made of hops. So I'm not going to tell what it is, but he is actually a connoisseur of different hop juices. He likes hiking, reading podcasts as well, and he will talk more about who he is and what he is here to do. Jonathan?
Thanks, Juanito. Good afternoon, everyone. My name is Jonathan Cardenas. I'm a graduate research assistant at CEA Lab under supervision of Dr. Ferrarezi, of course. And welcome to you all to my seminar presentation. Thank you all for being here, either remotely or in person. So let's begin with the presentation.
Just click on it once. Click it, yeah.
Actually, it's the computer. There we go. So, well, Juanito mentioned almost all about myself, but I'm originally from Ecuador, as he mentioned. I left my country after finishing with my undergraduate studies, and I had the chance to go to work to Czech Republic for 2 years, where I got involved and I got familiarized with remote sensing and plant phenotyping, especially from a fundamental perspective. They were developing cultivars for stress resistance, or developing cultivars with potential as a stress varieties resistant to stress. But what really is plant phenotyping and how is it connected with remote sensing? So essentially, plant phenotyping is measuring specific plant traits to describe either the functional or the structured part of a plant non-destructively using different or a wide range of imaging sensors, and they want to obtain a quantitative description of the plant phenotype with the purpose of speeding up the understanding of the genotype to phenotype relation. This from a fundamental point of view. However, phenotyping has opened new opportunities for productive point of view, and new opportunities not just for remote sensing, but also for computer vision, we are currently using computer vision for automatic pest and disease detection. We are using it for evaluation of good quality in a non-destructive way. And of course, we're using it for yield prediction. However, there is a drawback, especially on outdoor conditions, that the implementation could be quite challenging because of the cluttered environments that we could encounter in outdoors. And we could have a challenging image processing involved, and we could have a high level of difficulty in extracting individual plant traits. That's why, luckily, we are working with controlled environment agriculture in our lab, and this could be one ideal setting for remote sensing. Why? Because as you can see from those images, we have uniformity across a production setup in general. We have less environmental noise, meaning that we have environmental conditions more controlled, giving us an easier way to extract high quality plant traits. And it's a really suitable environment for plant modeling, since we are able to gather information from multiple sensors, not just imaging sensors, but also environmental sensors. However, there's another challenge when we want to put together all that information, and it's that the multiple remote sensing approach would create a high volume of data that at the end of the day could be difficult to interpret. Then the vast majority of the crops being grown in CEA facilities are leafy greens. And the way that we measure the yield with leafy greens is basically counting leaves and stems being produced, considering that's our end product. So we consider the shoot biomass accumulation as a proxy for the yield, right? And at an individual level, it could be a straightforward measurement, accurate. However, it's destructive and it's time consuming. Either to measure leaf area, fresh weight, dry weight, and it's even more challenging when we think of grown parameters at the large production scale to be used as descriptors for yield. That's why I consider remote sensing as a really useful tool or a combination of description of plant morphology and physiology that would let us estimate accurately the yield in leafy greens. But we are gathering so much information that we will run into the problem of lack of interpretation, and that's why we think that machine learning could be a potential tool for us to discover patterns in between so many large data sets that we would obtain. Then we can have certain level of flexibility. I will tell you why a certain level of flexibility later to adapt to new data. And rather than considering and focusing on a single trait, we can make trait association and consider multiple traits simultaneously. That's why with my project, we have hypothesized that the remote sensing platform that we will develop will extract geometrical plant features that will allow a machine learning model to estimate accurately the biomass accumulation in lettuce specifically as a leafy green, and associate thermal and spectral features as a tool of early stress detection. We have divided the whole project in 2 chapters that we have organized in multiple experiments, but specifically for the first chapter, we want to combine all the technologies that we have chosen ahead and integrate all those technologies by a consistent imaging processing pipeline or a workflow that would be capable of generating robust plant traits. So we start with gathering high quality information, those robust plant traits, and then we try to train an adaptable machine learning model to predict lettuce biomass accumulation using the most representative geometrical traits. So we are using just one of our remote sensing technologies until chapter 1. For our second chapter, we want to validate the additional features that we could extract from the other 2 remote sensing sensors. We have the thermal and spectral sensors that are going to be used, or we will going to validate their use as physiological indicators of the stress. Then to try the performance of the model we build, we want to try the performance or the model's prediction performance across different treatments using an abiotic stress severity experiment. So as I mentioned before, for my chapter 1, it's oriented more towards putting all those sensor technologies together and starting to train our model. So for this, we have selected 3 different remote sensing principles. First, starting with color RGB and depth camera, that besides giving us a color description of the plant or the common color information that we can gather from a normal picture, giving us information in the R, G, and D channel, which is the description of the color. It adds an additional channel that we can see here as the depth information, which is basically the information of the distance of each individual pixel in the image from the object that we're focusing to the camera. That would give us the option to calculate the plant height and build a more robust geometrical description of the plant. Our second sensor technology would be a thermal infrared sensor. With this, we want to calculate the temperature of each individual value inside of a picture. And with this, we want to get a proxy of transpiration rate, because we will obtain, or we will try to obtain, a good description of the leaf temperature. Then I would also like to take the chance to thanks to one of my committee members, Dr. Van Yersel, who actually he shared his knowledge about filters, microcomputers, and spectroscopy. And in fact, this is what we did on our third remote sensing technology. We basically put together a microcontroller, we put together a near sensitive camera, and we put together a dual bandpass filter. What we want to do with this, we want to obtain a really common and well-known index, which is the NDVI. And with the dual bandpass filter, what we are trying to do is we are trying to just let pass to the camera lens 2 wavelengths of light, being the near infrared and the red spectrum from the visible spectrum. So that's how we will isolate, or we will try to narrow down the wavelengths for us to be able to calculate directly the spectral index that we're interested in. Then really important as well, I would like to describe the general experimental setup that we will have in across our experiments, starting with the growing system. We have chosen a deep water culture system that we have a fixed volume of water inside of a tray. And that fixed volume of water is being aerated by a pump, so we can keep ideal oxygen conditions for root growth. Besides, we have put on top of each nutrient solution in each tray polyester board that we are going to use as the support for our net pots and substrate where our plants are going to be growing. So as you can see on the right, on the picture on the right, we're going to have a total of 6 trays distributed along our bench length, which is going to be 50 feet length bench. And each individual tray will represent a treatment. So if we get closer to an individual tray, in our polyester board, we will have a space to grow 55 plants per tray. And in total, for our 6 trays or 6 treatments, we're going to have a total of 330 plants per experiment. Besides that, something that it's really important to consider here, it's our positioning system set. So with positioning system, I mean that we want to replicate the same space distribution for each tray that is going to be our treatment. In fact, we want to obtain the same exact picture across all the bench, so we can replicate the same image analysis in each individual tray. Remember that we were using the board or the polyester board as a support for the plants, but besides, we want to use it as a contrasting color reference that will help us with the identification of each individual plant in the pictures. So as I told you before, we want to take advantage also of the greenhouse infrastructure, so we can mount our plants on top of the bench, and we can have the multiple images of each individual tray. Okay, so I'm summarizing in this framework how we are gonna organized the first experiment for our chapter 1. And in fact, the experiment is running right now, and we have decided to run a contrasting nutritional rates experiment. Basically, across our treatments, we're going to have 3 different rates of nutrient solution, and our objective with this is to have a population of heterogeneous plant sizes. We are doing this because we want the system to be fitted with multiple plant sizes in order to avoid overfitting, meaning that it will perform like wrong predictions based on just a small group of samples. That's why we are trying to generate an heterogeneous plant sizes all across the population. So as you can see here, we are doing 3 harvests along the whole crop cycle. And we are taking in each harvest the manual measurements for leaf fresh weight, leaf dry weight, and leaf area. And besides, we're going to be imaging 3 times a week. Of course, what we will need to correlate later are the images from the day that we did the harvest. And then we will also, with this experiment, try to see where are the flaws in our imaging processing and standardization. That actually, for us to have a good performance of our machine learning model, we first need to make sure the quality of the features that we're extracting from those images. So here you have like some preliminary results. Here on the left, you have the image processing workflow that I'm following. And I've been doing it in Python. There's a good open source library called PlantCV that it's available for plant phenotyping. And if you're familiarized with Python, it's not so complicated to use. They have actually summarized properly like the most useful functions in image analysis, but applied into plant analysis. And through that processing workflow, we have been able to obtain the geometrical traits description at a trait level. There you have the distributions for each specific geometrical trait. And then we can also obtain information at a plant level. We can have, in this example, we can see the count of pixels or the area for each individual plant, giving us this high throughput approach that we want to achieve. Here you have actually, this was the last image that we gathered this morning, and how I'm usually doing the imaging of the plants at the greenhouse. I really want to emphasize in how I will perform the machine learning training phase. So please remember what I told you about the first experiment. And let's, for instance, suppose that my aim is to predict the biomass as fresh weight. So that's basically the variable that we want to predict. And from the image analysis, we're going to gather all the image geometrical features that we can, and we will select according to the correlation or the representative in relation to the fresh weight that we want to predict. We will select a few of those geometrical features that will be known as our potential predictors. So we are not using all those geometrical features because probably not all of them will correlate with our fresh weight, but we will create a matrix of predictors that will allow us to fit a model. So that as our first step. Then we will recall, or we will take the data that we obtain in every of our harvests, the fresh weight that we obtain mainly, and we call them labels. So it's basically the real value of our prediction, or what we want to achieve with our prediction. This method in machine learning, it's called supervised learning. And what we are trying to do is predict an expected output, in this case, the fresh weight that we obtain during our harvests, giving a set of inputs that in case would be the predictors that we chose from our image analysis. And just a clarification there, our labels are nothing more than the desired prediction that we want to achieve, or the value that we're looking for. So after choosing both the inputs, being the potential predictors, and our output, that is the fresh weight, we choose the best regression model, a regression model because we're measuring a numerical parameter in terms of grams of fresh weight that will yield the best performance. So for our second chapter now, going forward with the with the other remote sensing technologies that we put together, we want to test the model performance first of how our geometrical features were able to predict or estimate the biomass accumulation. And we also try to give an additional capability to the platform and is an early stress detection. So as you recall, I told you that we are also adding a thermal infrared sensor. Basically, from the leaf energy balance, we know that at a higher transpiration rate, we will have a cooling effect around the leaf, and the leaf temperature or the temperature around the leaf could drop. However, the opposite is also possible, and that's what we will try to quantify with the thermal infrared technology. And for this, or to validate the values that we could get with the camera, we want to use an induced plant water deficit experiment, where we will use polyethylene glycol to simulate the plant water deficit inside of hydroponic conditions. And then we want to take all those raw temperature values per pixel, and we want to normalize them according to the conditions that we will have inside of our greenhouse. So we don't want to use like the raw readings of the temperature, because that's not going to be accurate. We need to normalize according to the environment where we are growing our plants. Then regarding the spectral reflectance, we need to keep in mind that the most accurate way of measuring reflectance is through multispectral cameras. Basically, the value that a multispectral camera will be throwing, that was going to be our NDVI, the NDVI ground truth, or the value that we will be comparing the values we obtain with our camera. And let's remember as well that the light reflectance in the visible region, especially in the red, it's highly dependable on the concentration of chlorophyll. So we would also like to run an experiment where we are comparing, once we validate, of course, that the way that we are obtaining the NDVI through our methodology correlates well with what our ground truth will be. So later, to try to obtain a way of getting leaf concentration or NDVI as a proxy for leaf concentration, we want to correlate SPAD readings with the NDVI index values that we will obtain for each individual plant. So after the validation is done, we want to choose a plant stressor, one of the most common stressors that we will encounter at the CEA facility, either being the EC levels or different light levels, and we will run at different stress severities per tray inside of our growing bench. Then from our validation, we know that we have additional features which are quantitative stress indicators. For instance, it could be an increase in temperature or a lower NDVI values that could show you or could show us a pattern or a way of characterizing a stress pattern by putting together the geometrical description, the thermal description, and the spectral description. Actually, one of the points that we wanted to emphasize on the high throughput is that sometimes the stress is being detected not in a single time point, but across a trend. So that's what we want to achieve, like also control time series where we can see the change in a specific feature that we are measuring. So as you can see on the right, that's basically the same matrix that we constructed with our predictor or our predictors coming from the geometrical features, and we want to add the features coming from the thermal imaging and from the NDVI or the spectral sensor, in order for us to be able to detect the stress based on the association of all those features. Then, as expected outcomes for each chapter, on the first chapter, we hope to achieve a remote sensing platform that can extract efficiently multiple geometrical plant features from each individual plant. And then we want to construct with those geometrical features or descriptors a robust machine learning prediction model that would estimate accurately the lettuce biomass accumulation. Then for our second chapter, we expect to show that the performance of the model was acceptable in new instances or in new treatments, in new situations, proving that the model could be used on a routine basis to predict plant biomass accumulation. Then we want the features from the thermal and the spectral sensors to be validated and to be used, or hopefully that will be used as indicators to characterize those stress patterns a long time. And then this is important with the certain level of flexibility that the machine learning model could offer us. And our same model, the same model that we will develop, is not going to be used by another species of leafy greens or even a different lettuce cultivar. However, we would like to create a model methodology or a framework that could be easily replicated to adapt to more lettuce cultivars or another leafy greens species. That's all from my side. Thank you all, and I'm open to any questions. Before we go with the questions, I just wanted to thanks all the people from the Horticultural Physiology Lab. They have been either helping me with learning new things about image processing, filters, microcomputers, 3D printing. So, and I would also like to thank actually these teams. I'm not in communication with them, but I've been using their Python package, and it's extremely useful. They have made a huge development in terms of plant phenotyping at a low cost. Thank you all. Questions?
Are you gonna have a whole pile of cameras, or are you gonna move the cameras over the plants, or are you gonna move the plants under the cameras?
No, actually, let me show you with the images. So here we have, basically we have like 4 of those holders on top of each tray that it basically has exactly the same height. The trays are exactly like distributed, so we can have underneath, or the field of view of the camera will cover each individual tray when we are mounting and changing the camera place. So it's basically the camera moving along the bench.
Yeah.
I mean, if it would be an engineering research project as well, we would automatize like the way like the camera.
Aren't you an engineer? Wait a minute.
That's what he wanted to do, but I had to remind him that this is the department.
What about the? How much will it cost?
If you go back to the RGB image,
That's true.
the slide, yeah. Go here. You have to take RGB plus depth, the deep. Is this spatial camera
Oh, I know.
or?
Yeah, let me explain further with so yeah, it's actually, I don't know if you guys remember this Kinect that once it was like a video game where you could like move and play jumping or something. So it's basically like the third version of that device, and that it's like a depth sensor that it basically measures the distance. Let's say the camera is here.
Okay.
It can measure through a near infrared beam of light. It measures the time that the light or the time that the light takes to bounce back to the camera when it hits the object. And that gives you an estimation of the distance in millimeters. So here you have like the RGB, but through the processing, we have just here the image that it's showing specifically the 4th channel, that it's the depth channel.
Oh,
So
okay.
here, each individual pixel in this image is telling you value in millimeters and how far that pixel is from the camera on top. So another advantage of the polyester board that we are using is that's our ground reference for the distance. And then we have the distance of the camera to the plant canopy. And we'll just like take the difference and we can
Calculate the height.
almost accurately predict the height of the plant.
So this is spatial camera. It's not a regular camera.
No, it's not a regular camera. It actually has a depth sensor, which is that beam of near infrared light that it's sending to the object that it's focusing.
You're using it.
Difficult to get.
So you're using it primarily to get the plant height, not the plant volume.
No, actually, what we're getting, or the way that we want to give the whole geometrical description is through the RGB, the normal camera. We are going to extract the plant length, the plant width, and then combine what we will retrieve with the depth information or from the depth camera to give like all the description or the volume description, put together all the geometrical variables. Which in fact is another advantage of using a machine learning system that usually it will take all the, let's say, all the features that we extract, and it could also give you associations of features combined together that would let the model improve the performance.
How much does this gadget cost?
It's around $399.
It's not too bad, plus there's.
And probably there are even cheaper devices now.
So hopefully we can get rid of measuring plant height manually. At the end.
But I mean,
We can lower this to the rest of the department.
And leaf area, right?
And leave area too.
Well, leaf area is easy, but it's very, very reasonable. High you can get a 4 channel. It would be very accurate. What did you have to check how accuracy?
it's really accurate. I mean,
Accurate, right?
I took until now, I actually got let me show you. So I put the camera on top of the polystyrene board. It's around 124 centimeters, and it's giving me like 1,245 millimeters or something like that. So it's really accurate, and it keeps like the value along the board. So yeah, and actually what sometimes when you are measuring manually the plant height, you don't know exactly like which is the point that you will take as a reference for the top of the plant. You don't know exactly what is like the highest leaf. However, here we would like take the highest point in that area where the plant is,
Growing point.
that you know that it's gonna be for sure, or if it's over that 124 centimeters of the raft, that's gonna be like your plant height. You know approximately like how much the value is gonna be.
You mentioned about the 4 channels. You said I have 4 channels. You have a camera, more channel, it's more accurate. Like 5 channel would be.
Oh, no, actually, the 4th channel, it refers to the additional depth channel that it's adding to the image form. So it basically, the 4th channel will be used to keep those values in millimeters, besides the RGB, which are the
Oh, okay.
the reflection in those colors in the image.
Okay, that's a different channel.
Any other question?
I have a question, like, did you ever comparison your research, like, if I'm talking about the leaf area, then did you ever comparison with the device who's calculating leaf area meter? Then with your camera, did you get any difference between it?
Well, actually, we are not able to calculate the leaf area meter with because that's the objective of the model.
No, just the difference, like, did you see any difference or it is accurate?
He hasn't developed.
No, I mean, because we want to, that's what we want to achieve, basically. Like the leaf area meter is giving you already the leaf area value of the plant that you are like passing through, right? But what we want to achieve is just take every individual, let's say, a dimension of the plant or a geometrical description, plant length, plant width, plant height, and try to get a model that will tell you, oh, with those predictors, you will have this leaf area. Which that's the most challenging. Maybe leaf fresh weight and leaf dry weight are not as challenging as the leaf area, because actually the value of the leaf area meter, the value that is giving you, you are like putting flat leaves and passing through, right? So it's like quite difficult to get that. So that's one of the challenges, actually, like get an accurate leaf area.
Will you validate that over the growing period?
Yes, for sure.
Like,
For sure.
how often are you gonna do it?
So lately I've been validating every week, every time that I have a harvest. And there's a funny story actually behind the validation, because you could validate in 2 ways. Taking yourself the measurements, like the plant width, the plant height, the plant length, or taking image J and zooming into the plant, and like how like to put a line across the plant to know like what's the horizontal distance or the vertical distance. And that's a better method of validating that you doing yourself, measuring by hand, because it's a high, depending on how you measure as well. That's really variable. Wait,
But
I'm
you will
using
run through like an actual leaf area,
oh yeah.
right? Yeah, okay,
Mhm,
yeah,
mhm.
yeah.
Actually, that's in every harvest, we are getting leaf area, leaf fresh weight, and leaf dry weight. And that leaf area, we are doing it with the leaf area meter. Mhm. Anything else?
What cultivar are you using?
Using Chicarita. It's a romaine lettuce cultivar.
Why'd you pick that cultivar?
So I picked it, I saw the pattern that growing more like vertically, right? And it's keeping the consistency along like the vertical growth. Besides, since it's not going too much to the sides, that tackled down one of my complications, which is the leaf overlap. And that's another intention of the way we are harvesting the plants, that we are leaving, let me see, it's here. So you can see here that you have now empty spots here in between plants. So when I'll be done with the second harvest, I will leave empty spots all across the raft for me to avoid that overlap and try to get like a more or a better description of each geometrical feature. And that's a challenge to tackle down as well. That leaf overlap that we could have, that's real in fact. That's how producers grow their plants.
What is the production cycle? How many days?
It's around 30 to 35 days. I'll probably do lower, considering the overlapping issue, but I need to see if along that 5 days difference, I would have overlap or not.
Jonathan, assuming everything goes well as planned, so how difficult would it be to actually extrapolate this setup for other crops? So we have a lot of people working different crops that will benefit from this without having to do everything again from scratch. What would be your
Well, that's in fact a challenging question, and that's why I like got that last outcome that we could get, like to just set a model framework on how to do this in an easier way, like not go through which are the features that I need to extract, but also develop a system that could already extract the useful features, not go through the process of training again. But it's confusing because at the end of the day, you will need to train again. In fact, in our project, the constant iteration or adding new values of different way of the plants growing in different greenhouse would give us a better performance to generalize. So it's a constant iteration of adding constantly information to improve the single system.
Any questions from one time?
I think we might have time for one more question.
Yeah.
Can you check?
Someone online has any question?
Click on participants. All right, so I think if we don't have further questions, can we give Jonathan a big round of applause?